All right, let's start talking about Agarose Gel Electrophoresis, right? Allows us to separate DNA, RNA, and proteins according to their size. So essentially using an electric field, we know that nucleic acids, well, maybe we don't know, and proteins are negatively charged. And so they will, they're attracted to positive charge. So if you run an electric field through, you will see the molecules move. And we're using an agarose gel, and agarose is very, very similar, has the same stuff in it, agar, as Jello. And so you can think of it kind of, it looks like a solid, but you know, it's kind of softy. So it's a matrix. And so if you think about it, something really thick, small fragments can get through quicker than big ones that will be more retarded by the gel. So this is just a little telling us exactly what we need, right? I'm not going to read that to us. Again, this is just a protocol from a company because I just stole this and it had some nice information on it. So I posted it and it's in our little talk today. So it just talks about how, right, agarose gel appears to be totally solid, but blah, 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 the little holes come through. So what they're showing us is in A, here's our agarose gel and the wells, which are little depressions, kind of like little holes, rectangular shaped holes down into that gel where you would add your DNA sample. Now, we know when we talked about setting up PCR, we're essentially pipetting clear liquids into a tube. Now, our GoTac green actually has loading dye in it, so that helps us. And then we usually add loading dye to our samples, and that not only helps color it so we can see that it got in the wells, but it has glycerol in it, which makes it heavier. Because otherwise, the gel is going to be submerged in liquid. If you try to squirt liquid in liquid, it won't sink to the bottom of the well. It'll just squirt out and there goes your DNA just in the buffer. So we add the DNA to the wells and then we apply that electrical charge, negative to positive. And so our sample will run out of the wells, cutting through that gel and separated by size. So the guys down here are the smallest pieces and the guys up here are the largest. And if we have a molecular weight marker, also known as a ladder, of known sizes of DNA, then we can run that. So that would be like usually in your first lane over here. And you could say, you might have a piece that's 10,000, 8,000, 5,000, right? 1,000, 500, 300 base pairs. And so then if you're going to pretend like, let's say that guy's your 300, it's the smallest, then it goes 500 and then 1,000. So we'll say this guy right there is that 1,000. In the, sorry, I'm doing the wrong side. Hang on. I'm pointing to, here's our molecular weight marker on this side. I just said it was in the first lane. So that bottom guy will pretend was 500. Is that what I wrote over there? And then we had 1,000 and then we had 5,000. So this, whoops, that guy right there is our 5,000. And so then when we run, if that's our 500, if we run our sample in this lane B, then we can say anything between that 5,000 and that 500 has to be between 5,000 and 500. And so we could guess that that guy right there might be 2,000, right? Because if our molecular marker in between this 500, that guy right there is at the 1,000, then you have an approximation of what the size of your fragment is. So you always choose your molecular weight marker, your ladder, based on what kind of fragments you're looking for. And so there's a whole bunch for sale, and that's how you can determine whether you're in the right ballpark of size for your PCR product. And so we need to take that into consideration. What kind of ladder are you going to choose? What fragment size are we looking for? That was our SNAP gene. Putting our primers in our SNAP gene, and also, right, our PCR primers in Primer Blast just told us what our size is going to be. And then we also have to decide what percent gel to make. So if you make a higher percentage gel, it's going to be thicker. There's going to be, it's going to be harder for fragments to get through. So big ones might not even get through at all. So we have to pick the appropriate percentage gel for whatever sizes that we're looking for. So that's sort of what we're going to talk about today. And remember, again, we'll talk about this, but we can't see the DNA on the gel. We can only see the dyes that we put in the wells, but we can't see the DNA until we stain it with something that intercalates and fluoresces or is an actual dye that you can visualize with the naked eye. So that's what's going to happen today. We're going to talk about gel electrophoresis and all the things we need to do in order to have a gel and run one, right? We're going to pretend like we did PCR. We have our little tube. It looks green because we put the GoTech green in it. It's out of the PCR machine. Now we want to resolve and see if we got the fragment, a PCR product, the size that we were expecting. So what would we need to do? Well, we have to make agarose. We have to make the gel, and then we have to pour it, right? And then we have to let it sit until it's all ready. And so that's just with this little schematic. Here we are making buffer. So we don't run it in water. We have to run it in a buffer. And that is because it needs to have buffering capacity when you run electricity through it. So if you remember back to chemistry and buffers, which I remember hating, I just buy it. You know, you need those ions in there to make sure that current is constant and is even throughout your gel. So you're just going to take my word for it. You need a buffer. And generally we store these buffers at a concentrated percent, and we make it 1% when we're ready. And then we have to make whatever percent agarose gel. So we need a percent agarose gel. We need this to be end up at 1x. What do we have to do to Jello to make sure it goes into solution and then ends up setting as a gel versus liquid? Well, what do? First, we add the agarose, we swirl, and that's going to look like a slurry. It's not going to dissolve. So it's not going to look like pouring salt or sugar in water, where the more you swirl, it'll eventually completely dissolve. You can still see the little chunks in there. You want to make sure they're nice and even. And then we stick it in our microwave and we zap it. So just like you'd make for Jello, you have to, the molecules need to come up to boiling or very close to boiling in order to then, when they cool, to set as a gel versus staying as the slurry in a liquid. And so we zap it, usually start with 1 minute, depends on how much you're making, swirl a little bit, another 20 seconds, swirl a little bit. You have to be really, really careful when you're making agarose in a microwave not to leave it in too long. There's something called superheating. So if you superheat this kind of solution, it won't boil, especially if it's in a flask with a smaller opening here and wider down here. It will superheat, and when you touch it or move it to swirl, to mix, it explodes into, it doesn't explode, it just boils, right? That activates that boiling and it shoots out the top. So the last thing you want is that to shoot out the top at your face or your body. So anytime we pull anything out of the microwave like that, we always swirl a little bit away from ourselves and away from anybody else. And we microwave at short intervals so that it doesn't get to that superheated extent. And then you're supposed to let it to cool to about 60 degrees Celsius. There are labs who have a 60-degree-Celsius water bath that you then stick your flask in, so it can't drop below 60 degrees, right? If you're going to go out, have lunch or do something and forget about it. You don't want it to get coagulated. You don't want it to set into that gel in your flask. If it does, you can always re-microwave it. It just takes that much longer. And then you got to swirl and microwave, swirl and microwave. And so when it's about 60 degrees, you down to 60 degrees. If it's boiling, it can bend your well and bend the plastic of the actual tray. So we put the tray together. So we put the little dams on the end. Old school, you just put a piece of tape across there because you lost the dam part, the little thing that blocks it. And then you put your comb with your wells so that you can actually have someplace to put your agarose into. And then you pour into this guy. It says wait 20 minutes, and then you can pull off the edges, pull out your comb, and you have that nice solid, gel. And then it's got the little holes where you can put your sample into. And so it says wait 20 minutes. It depends on if you have a really big gel, mini gel, yes. If you have a big gel, it'll take longer. If you really want to hurry it up, you can actually pour in a refrigerator to make it cool down much, much faster. So that's our overview of what we need to do. And so now we are going to do a little bit of work, and we're going to, oh, that guy didn't come with me. Well, I'll move that guy. Okay, so let's bring this down here, because we want to remember this stuff, but we don't need it. This guy, yeah, I can move that guy. Okay, so we're going to make, so we want 1x right at the end. That's what we want down here. And we're going to make 100 mils of 1x. We're just making that up, right? If mini gels usually take like 30 or so. The bigger gels take more. If you make extra, then you can just microwave it for next time, as long as you have something with a lid. So you could make it in a glass bottle and then put the lid back on. You don't want to re-microwave agarose too many times, or it becomes more and more concentrated every time you boil it and you evaporate off some water. The 1x becomes a little more concentrated of your buffer, and the agarose might change from 1% to 1.05% or something. So you don't want to, you can microwave it a couple times, but you don't want to microwave it 100 times. Okay, so what do we have? We're going to say, well, we're not going to say, they're telling us we have 50x buffer, and then we have water, and it's going to be your guys' job in a breakout room, randomly selected pairs or so, you want to make a 1x buffer, 100 mils of a 1x buffer from a 50x concentrated buffer. So that is your job. So I'm going to make some breakout rooms. And we are going to say there's going to be about 2 of you per room. And we're going to go. You guys are going to do that equation. And then when you're done, just jump back into the main room. So 1x, 100 mils of 1x buffer. You have 50x concentrated buffer. So tell me how much buffer, how much of that 50x buffer, and how much water you're going to need to add. And we are going to, I guess okay, resume recording, right? So here we are, we've got our equations out and how we work through them. And again, just this second one was in case, right, you want to make something else. 1x, 30 mils of a 1x solution, same exact setup, just different numbers. Again, if you wanted this to be 2x, this would change. If you started with 10x, that number changes, but the setup is the same. Okay, let's see what comes. Okay, that was just the numbers down there. Okay. And the words. Oh, all right, sorry. Okay, so now let's talk about the actual agarose. So we made the buffer. We said we wanted 1x whatever buffer. TBE is what we usually use. But let's talk about how do we know how much agarose to make, right? We didn't talk about that yet. We just showed the little agarose guy sitting there. We put some in the 1x TBE and then we do the microwaving stuff. So what do we need? How much do we need? Generically speaking, 1%, 1% agarose is usually the safest if you don't know anything about what you're looking for, but you really should anticipate the size of your fragment so that you can choose a percentage that will resolve that fragment the best you can. And we also need to know for our PCR our misc priming. So what other possible PCR products could come out with these primers? And that's why we did that primer blast check. So if we were remembering back to my little example, my PCR product, I think it was 512 base pairs, 513, something like that. So that's the size I want to see. And I want that fragment to not be way at the bottom of the gel. I want that to be a nice clean band, nicely resolved on my gel. And then if you remember, I have my sheet, which I forgot to show Wednesday, which I can show you guys. But if you remember when we went through my example, I think I had 1200 base pair possible mismatch. There may have been like a 900, 934, something like that. And then I think the rest of mine were 3000 and 4000 base pairs. Okay, so these are all the possibles if my primers sit in the wrong place. Like, so my annealing temperature is a little bit too low, right? Remember we talked about that when we talked about PCR. That's what controls for where it sits. So I want to see this 512. And if I see other bands, as long as they're not right next to crowding my guy, I don't really care. As long as I can be sure I have that 512. So we would definitely use a ladder and we'd pick a molecular weight marker that resolves around that 500. We wouldn't want one that's like just a 1 kb marker, that the smallest marker is 1,000 base pairs, 2,000, 3,000, 4,000, 5,000, because that's not going to tell me anything down there. I want ones that maybe have a 300 base pair, a 500, an 800, 1,000, a 1,200, a 2,000, 5,000, 10,000, something like that. And so that we just look in a catalog and we just buy one, or we dig through the freezer and we just pick the one, right? So, but we have to know how much agarose, what percent agarose we're going to make. And if it's going to resolve. So we're going to use this top guy. And again, I'm sure this is online. This is actually what I had printed out forever ago. And this was taped onto my lab bench when I was a graduate student, because you constantly are making all different types of gels if you're doing molecular biology and a lot of cloning. And I did a lot of cloning and I worked with a lot of small pieces of DNA. So if you're working with just regular old fragments that aren't small, too small, we would be using just a regular Agarose LE. This is a certain company's brand, but just your standard molecular grade Agarose. And we generally run diagnostic gels in 1x TBE. Again, that's that buffer. 1x TAE is another buffer. It's tris acetate EDTA. This is tris borate EDTA. TBE is a little better of a buffer, has a better buffering capacity. You can run a gel longer and still have it nice and smooth. TAE doesn't have as great a buffering capacity, but generally we're not running stuff so long that it would matter. Resolves a little bit cleaner here, but we would use tris acetate EDTA if you're trying to extract a band from a gel, or if you're transferring it. So just looking to see if you have what you need. We usually just use TBE. So that's the key, legend, I don't know what to call it, that we would use. And so over here, this is the base pairs that it essentially can resolve. So this Agarose is not recommended for pieces that are smaller than 300 base pairs. It just doesn't have the resolving capacity. If you need that, then there's something called Metaphor Agarose, and this is a specialty Agarose. So instead of having to use acrylamide for small stuff, you can use an Agarose gel. So I use this a lot. I cloned a lot of things that were 150 base pairs, 50 base pairs, 200 base pairs. And so I needed to really see well that I had the right thing. And so I almost always ran a 3% gel because I was always looking for stuff in there. That's why that arrow is there. Again, this was just stuffed onto my, it's got some posterity, nostalgia, on my bench. And then down here is the dye chart. This happens to be for Metaphor because that's what I almost always ran. And that just tells you the loading dyes that we add. Now the green, I don't know where that runs in GoTech green. But if we made our own loading dye, and then there's also orange G, that's an orange color that runs smaller. So you can have 3 different colors in your gel, and this tells you where they run in TBE and where they'd run in TAE and what percent agarose. So like, for example, I used to run this 3% TBE. And so if I had my gel, and here's my wells, right? And we're going this way. The dye actually travels by size as well. It's just a pigment, right? And so it has to get through those little holes in the gel, just like a DNA or an RNA fragment. And so the purple runs as a dye front, and then orange would be down here. Orange runs really small. And then I don't, do we have a turquoise? Yeah. Would run up here. So we'd usually try to trisect the gel. That way we know we're not running our fragments off the other end. Remember that gels are in, not water, in the 1x-TBE liquid. And so if DNA travels and you leave this on, you leave the electrical field on, you forget about it, the dyes will run off into the buffer, and the fragments of DNA will run off into the buffer also. So you can't just let a gel run indefinitely against all your DNA. We say it dives off your gel into the swimming pool of buffer, and then you have to start over. Okay, but I digress. So let's go back to this guy, how you would choose. So again, I'm down here at this 500 for mine. And it, you know, that says between 500 and 4,000 is a 1.25% gel. 300 to 3,000, I don't need anything smaller than 500. So they're just telling us that you'll see that 500, but you won't necessarily be able to tell the difference between 300, 400, or 500 base pair piece. Those are all going to run right together. I don't care, right? We are expecting nothing down there. Primer dimers will be a lot smaller, so they're not going to interfere. We'll talk about those when we actually look at gels. And the rest of mine are bigger pieces. So as long as we resolve away, 1.25 should be fine. Anything more than a 1.25% gel is going to take forever to run. And so at that point, I would switch down and do a Metaphor gel if I really wanted to see small stuff, right? And resolve down there. But we're going to say, I'm going to use that 1.25% gel. So you guys need to go look at your fragments and decide what percent gel you guys want to make. So I want to make a 1.25% gel. So I am going to come to this. So I want, what do I want? 1.25% agarose, 1x-TBE, and I want to make, let's make it easy for our first one. Let's make 100 mils. Okay, so I want you guys to pull up your mispriming so you can write down, like I did on the other slide, what all your possible, well, maybe not all, but at least the main possible mismatches. Now, it's possible it didn't list anything other than your gene, right? Your primer blast check should have found your gene, which may have multiple different clones and uploads into the Human Genome Project at NCBI. But if there's any of them that misprimed at other sizes, and you need to know what size you are looking for. So you're going to find that right now and have that loaded, or make your little chart. You can make it on a piece of paper, you can make it anywhere, whatever. It doesn't have to be typed up and so forth, just so that you have it. And then while we're searching for that stuff, I'm just going to set up my equation to figure out percent agarose. Now we can, like in lab, I always just kind of did it the cheaters method, which is fine. You can always do that too, but we'll have an equation so that we can always fall back on that also. Of course, I totally did it wrong Wednesday because I'd never used an equation. I always just did the cheater method in my head. So that was entertaining. I cut that out of the recording, so it's not in there. Okay, so before I send you guys away to go back to a breakout room and find that info, and then we'll come right back before you have to set up trying to figure out, you know, what percent agarose. We want to remember that 1 gram in 100 mils is always 1%. Remember, that was just our rule that was designated, right? Weight per volume. We're just designating, so whether it's salt, whether it's agarose, 1 gram of salt in 100 mils of water is 1% salt solution, right? 1 gram of sugar in 100 mils of water is a 1% sugar solution. That's just what it is, okay? So it works the same for agarose. So if you're making 100 mils, it's pretty easy, right? If 1 gram in 100 mils, right, is 1% here, then 1.25 grams in 100 mils has to be 1.25%. Okay, so I'm going to send you guys away and I'll set up my little equation while you're gone, and then we'll look at it. And hang on, I'm going to send you into different rooms. And so find that information, or if you can't find, you don't have it accessible, try to remember what your other sizes were, and then we'll come right back. Let's do the example first. How about that? Okay, so we were talking about 1 gram in 100 mils is 1%. That's the rules. That in turn means 1.2 grams in 100 mils is 1.25%. 0.8 grams in 100 mils, 0.8%, 5 grams in 100 mils, 5%, right? Anything in the 100, we got it. The problem is, what if we're not making 100? What if you can always, if it's up to you, you can make it always in 100 or 200 and, you know, do it the cheater method, or 50. And then all you have to do is figure out how much in 100 and then cut it in half. But what if you have to do 30 or 27 or some stupid number where it's not easy to do the cheater method? Then we have a cheater method for that too. So if you want to make, let's say we'll do 50, because then you can also do the cheater method to make sure that you understand why it's correct. And so let's do mine. Let's do the 1.25 guy right here. which we know is 1.25%. So if we know, so again, we're going to start with what we know, 1.25 grams in 100 mils, right? And we want that to be equivalent to X grams, we don't know, or question mark grams, right, in 30 mils. Okay, or whatever your end, oh shit, oops, I swore.
Oh, fuck.
Lily, can't say the F word. Recording. I forgot. I said I was going to do 50 mils to make it cheater method easy so we can check our work. And so now we're going to do math, and we have to use the math rules. And I don't remember what the rule's called, but it's where you can, right, as long as you do the same thing to both sides of an equation, it's totally fair game, and then each side is still equivalent. And so we want, because we want our X to be all by himself, right? So if we multiply both sides by 50 mils, right? And we multiply this side by 50 mils over 1, right? What do we get? Oh, I used the wrong color. Our 50 mils on this side cancels. And we end up with X. And remember, that's in grams. And then if we do this math, which let's do this math, make sure I'm not lying to you guys again. So 62.5 is 50 times 1.25 divided by 100 equals 0.625 grams. So that's how many grams we need to put in 50 mils to give us 1.25%. Now, if we use the cheater method, we can go back and say, okay, if it's 1.25 grams in 100 mils to give us 1.25%, if we have half the volume, as long as we do half of this and half of this, again, it's still going to equal 1.25. So let's do that. So half, right? We want 50. And what's half of 1.25 grams? Oh, 0.625, right? So that's how I would always do it in lab. I'd be like, okay, how much would I put in 100? Okay, if I'm only going to make 50, then I need half of 1.25. Oh, I want to make 200. I know 1.25 is 1.25% in 100. If I want to make 200, I just need twice that because I'm multiplying 100 by 2. It's just if you're making anything that's not an easy, 1/4, half. I mean, 30 is kind of a third, but not like a third is really 33%. So if you need some other form, like 30 mils, what do? So let's do this one that I have written down here. Let's do our 0.8% gel. Okay, and you want 30 mils of a 0.8% agarose gel. Quickie breakout to do that equation. Ready? Go.
Your life depends on it.
And we're back. Okay. So yes, we also had a suggestion to make it a little easier, which works too. Since it is, percent is the same as, in this case, as grams, right? You can just do the straight multiplication. You just have to make 0.8 be the percentage already, right? So if we did times, let's see, 30 times point. We have to move decimals. No, that's not right. If I knew how to turn decimals into percents, I would be all set here. .24, yeah, okay. So we would just do the straight math, 30 mils multiplied by 0.8%, right? So if we take 30 times 0.8 and we use percent, then that gives us, oops, 0.24 grams of agarose. And that should be essentially the same stupid math that I was doing. I hope, right?
Yep, it's the same.
Yay, see how we love math? Because you can do it so many ways. And then you just check to make sure it makes sense before you go with an answer. Right? Especially when I tell you to do an equation and I'm like, oh, I'm not right. Right? And so then if we do, oops, we do our canceling, right? We do our math on the other side. Right? It works that way too. So as long as you remember here, not to just do this multiplication, to make sure this is 0.8%. So you have to think about it like, right, it's 0.8 times percent and then multiplied by whatever volume, and that should work. Because otherwise you'll be off by 100-fold and you'll try to make some really thick agarose that'll never boil, and that really would superheat and become a total nightmare. Okay, so let's go back. Let me see. Should have had it in here twice, but of course I don't. Let's go back to this guy and just talk really quickly about what percent you guys think you should use of your gel for your PCR. So I know somebody's was, but it might be in our Wednesday group, had a pretty big PCR product, like 1,000 or more, or 900. Anybody in here? Or are we all smaller?
Mine is supposed to be around 1884.
Okay, so yours is big. So 1884, if we looked at this chart, this guy would tell us, right, a 0.7% gel. So in the real world, anything smaller than a 0.8% gel becomes really, really fragile. Because again, like if you think about, I don't know if anybody's ever made Jello jigglers versus regular Jello. The jigglers, all they do is just take out some of the water. So it's a much, it's not really harder. It's a much thicker Jello, right? So if you made Jello too thin, like you added too much water, like it would set, but it would be super jiggly and gooey, right? And that's the same way with a gel. So I would still recommend, just from experience, that even though this says do a 0.7, I would do a 0.8% for that, or at least a 0.75, because when we'll talk in just a second, you have to transfer that gel to stain it and then to look at it on a UV box. That's when you make a puzzle. and they're slippery, and the buffer's slippery. And that's when they fly out of your hand and splat on the floor and break into a bunch of pieces. And then you put it back together on the gel box because you still want to take a picture, hoping your band is there, not where it broke, so that you don't have to redo your experiment. So yeah, so even though your guy recommends 0.7, I would do a 0.8 for that. So anybody else? So if we're in the 1200 to 8,000 zone, they're telling us 0.85. So that's and again, 0.8 is pretty fragile too. So that you have to be more careful. So anytime you get a little bit thicker, it's a little bit stronger. It's less likely to fall apart while you're doing all the transferring. If you get too thick, then it takes forever to run the stuff through the gel just because it's so thick and you need more buffering and blah, blah, blah. So it's kind of annoying. So probably most of us are at this 1%, right? Or 1.2 or 1.25. 1.1, we could get crazy, right? Does that make sense? Anybody else? Nobody, does anybody have smaller than 500? We tried not to do that, but.
Mine is smaller.
Oh, how much?
423.
Okay, that's probably still okay. So in that case, I wouldn't go to like 1.75, but maybe I do like 1.3 or something like that. Again, if you were really trying to resolve it away from, like if there's a misprime at 500 or at 200, then we might want to get crazy and run something fancy. But it's probably fine if we did 1.25 or 1.3 or 1.5. Because again, it's not like it goes from taking 2 hours to 24 hours, right? Instead of a 1 hour runtime or 1.5, it'll be like 2 or 3. It's nothing to write home about. And then, yeah, it just really matters, to make sure you can resolve. So if there's pieces that are closer to you, to your size, you'd want to skew in the higher percentage versus lower percentage. So if you have, if you're the 1800 or 1830, sorry, I can't remember now what you said, but you also have some mismatches at 1500 and at 2000, then you'd be better off using a little higher, right, percentage gel, because then you make sure it resolves. Otherwise, they look like they're very similar in size because they're not resolved as well by that matrix. So that's just the games we would play to decide actually what kind of agarose gel we would make. And then we would go ahead and go through that protocol that we played with. Okay, so after all of that, okay, so let's go back here. And so now we have our gel, right? We took off those ends, we took out the comb. Sometimes you leave the comb in until you put the buffer over it, because occasionally, and it depends on humidity in the lab too, when you pull the comb out, the guy that makes those little wells, it can get stuck if it dried out at all and like pulled chunks out of the gel, and it could wreck your gel. So if you put the tray goes in this apparatus, and then the buffer goes in, and I don't know why they show it like that, because that's really stupid. You put it in this, and it's submerged a little bit over top, just enough to cover that gel. And then this is when you load it, right? This shows you, it's being loaded. It says you pour it in, but it doesn't, it's trying to show you. So this guy is in here, and it looks like this, but we're pipetting into these wells. Okay. So, and we're going to the positive cathode, I think it's called, versus the anode. I don't know. I can never remember those. The positive side, which they always color red, and the negative side, they always color black. And you guys don't have to worry unless you're really using something old in somebody's lab at CSUN, which probably still exists. But the safety ones all have a lid. And so the lid does not fit on backwards. Okay, so the only way, if you put your gel tray in backwards, so your wells are over on this side, then you could run your gel in the wrong direction. But in the good old days, you just had wires. There was no lids. And if you connect those wires and you turn it on and you stick your finger in that buffer, you will get the voltage of whatever you set. So if you're running 150 volts and you stick your finger in there, you're going to get 150 volts run through you, which is not good. It might kill you, and especially if you're doing, well, nobody does sequencing gels anymore, but we used to have to run like really high voltage through a sequencing gel, which if you put your fingers in it, it could actually kill you. And even those in the good old days had a protective top, so you couldn't necessarily turn it on and stick your hand in it. So don't ever stick your hand. If your lid's broken or you just have the leads because they can't find the lid, do not put your fingers in it. And so what you want to think about to make sure you didn't put the gel tray in backwards, because maybe somebody set this out to dry and they put it the back, you know, they put it in the opposite orientation that we want it. We always think of back to black and run to red. And so that means here's your wells. This is the back side of your gel, right? This is the way you want it to run, right? You want to go out of the wells, you want to go through that gel. So if you set it up backwards, let's just draw our little wells on this side. So let's say we had switched this guy backwards and you put the lid on and you run it, it's still going to run to red. And your DNA is going to go that way. And if you don't catch it in time, it will dive off into that buffer and be gone forever. And if you use your entire PCR product to load, you have to redo PCR and start over. And if you didn't, and you still have some left, that's great, except you still have to pour a new gel and wait and so forth. So you always want to make sure your back of the gel is to the black side and that we're running it to the red side. The other nice thing is now the boxes have timers on them and so forth. Again, in the good old days, there was no timers, you just turned it on. So we'd always set our timer for 5 minutes, go back and look at your gel and make sure you can see those dyes, that purple, that turquoise and the orange going into the gel in the right direction. So if you see them going the wrong way to the short side of the gel, stop, right? And then as long as you don't have to resolve like super, super fine stuff, you can set it correctly, even if it came out a little bit this way over on this side. If you then, you switch your leads or whatever, make sure it's running to red, like if you had them switched or the tray was backwards, then you can run it right back through those wells into your gel and not have to start over. So as long as you didn't dive them off the end, you can rescue that gel and just run them right back through those wells and into that gel. So that's nice. Let's see. Okay, and then that's just the words. And then this is just their little chart in case. And a lot of times we would write this up too, so you don't have to do the math every single time. It just tells you exactly for which, and this is for their apparatus. Like they tell you this one takes 300 mils, this one takes 400, this one takes 1,000, and then that's how you do the dilution, so that you don't have to M1V1 again. And then it also tells you, what voltage and how long it takes. So that's also nice to know. And they're showing you in a 0.8% agarose gel. So there's charts of that kind of stuff all over the place too. So you can guess. If you have no idea how long to run it for and you have to go to class, you don't want to run it too long. Again, you don't want it to dive off the end of the gel, and you don't want to run it not long enough, come back after 3 or 4 hours and have it gone nowhere. So it's nice to have a little bit of information about how long you should run stuff. Okay, and then we'll talk a little bit about looking at it. Remember, we talked about the different types of stains, right? You can't see the DNA running on the gel. You can see the dye lines. I'm going to post some gels. I meant to do it by today, but that didn't happen. And so that you can see what it looks like before staining and after staining, just the the dye colors running through versus once you've actually stained the DNA. And so this one, they're showing ethidium bromide. Ethidium bromide is the old standby. In order to visualize DNA, there's usually a compound that can intercalate into or in between the strands of DNA, at least in the major and minor grooves. And sometimes it can actually disrupt base pairing because it gets in there. And then it fluoresces if you put it on UV light. So this, you can see behind here, this is a little UV box. You put your little guy on there. You wear safety goggles and a cover for your face because UV, as you know, is very damaging to skin and eyes, can make you blind and give you sunburn and skin cancer. So you always want to have the protective gear and look at it for a short period of time. And so it fluoresces wherever there's bands of DNA. Here you can see those wells. That's that chunk of agarose. There's also something called CyberSafe, which stains it green. It's also an intercalating agent that also fluoresces. So they named it CyberSafe, but there wasn't really any information about how safe it was. So an intercalating agent by definition is a mutagen because it can intercalate and damage DNA. So CyberSafe, not so safe. So I don't recommend, I recommend treating it just like ethidium, gloves, goggles, don't get it on anything. And then there's other stains now that are just like a pigment dye. And so there's a blue stain, there's gel red, and they're not as sensitive. So if you have a really light band, you won't ever see it. But if you stain with ethidium, then you'd see it. But it's a nice option to use in the lab. That's at least as far as, it doesn't intercalate. So it literally stains DNA versus intercalating in and then having to fluoresce. So some of these, you need visible light to be able to see it, and some you don't. These guys are just showing their products from this company. So I'm not trying to, sell that. And in this case, it's a little piece of paper that has the ethidium soaked on it, and you stick it on there, and then you weigh it down and let it soak into the gel. And then you look at it on the UV to visualize those, to excite those guys and have them fluoresce. Ethidium is a hazardous waste, so you do have to follow protocols in the lab for hazardous waste, and it's specifically for ethidium. And so most people don't buy this method because you run all different size gels, and I'm assuming it's expensive. We usually use an ethidium bath just like you use here. And so in this case, the concentrated CyberSafe or concentrated ethidium bromide, a teeny tiny bit goes into your buffer. So it's a 1x TBE. You can also make it in water, but if it's in 1x TBE, when you slide the gel off of the tray, you don't want to soak the tray because you don't want the tray contaminated with any of that stuff, right? We only want to have whatever this little container, we use a reusable container that we use over and over and over, and you never touch it without gloves and, PPE. And then you put that stain in there, you wait 15, 20 hour, it depends on how much stuff you have in there. And then you can look at it on that UV light again. And so here they're showing us the different wells. This would be the molecular weight marker. This they're showing us as the negative, run to red, right? Run down to the positive. So the guys down here are the small fragments. The guys up here are the big fragments. So this is how we deal with ethidium also. And that's because then the only things that are contaminated with ethidium are whatever container you've made it in. And normally we don't even make it in a flask. We make it right in this container, right? We put the 1x TBE or water and a teeny tiny bit of ethidium in that. That way we don't have any flasks that are contaminated. We don't have trays that are contaminated. We don't have electrophoresis apparatus that are contaminated. Some people put this stuff in the gel and then everything's contaminated. So the flask that you made the gel is contaminated. All of that liquid in the buffer is contaminated, needs to be disposed of as a liquid waste. All the trays, all the apparatus, everything's contaminated with that mutagen. And so that's a big no-no for me. I know some labs do it. I just feel like it's super not safe. And so we're not allowed in the Malone lab to be crazy. Okay. And then their last is the same idea with their little Insta Blue. And so in this case, it takes forever because you're actually staining the DNA. It's not intercalating and fluorescing. All that pigment has to attach to it. So it just takes longer. And we still always use gloves and goggles and everything because any of these stains, we don't really know that much about them. So we always want to be careful, and we don't put them in the apparatus, and we don't soak the trays. So that's why when this guy's swimming in the Ethidium bath, the gel red bath, the cyber green bath, whatever it's in, he's slippery, and you got to get him out with your hand or a little spatula in your hand. And they just love to just shoot off your hands and jump onto the floor and become a puzzle. So you have to be super careful about that. And then this is just some pictures of actual, gels of stuff that they use for this company. So that's, just their little advertisement. And of course, I was going to collect all these pictures for you guys to show you, but of course I didn't in time because, that's how it goes. So I will get pictures of gels from people in my lab and post some stuff and make it. And there's a little video of somebody making Agarose. And I do suggest everybody make some Jello this weekend, right? Make it regular and make Jello jiggler. Okay. And that's yummy and it's fun. And then that'll give you an idea of exactly what happens when you put Agarose in the microwave. So that's what I have today for a Friday. I'm sure everybody is already exhausted and ready to go home, be done. I am going to stop recording.